Hello students, welcome to zoology class. Hope all of you are fine at your places. Today, in continuation to my presentations on cell structure and function, as I am discussing different cell organelles, today I will take next two cell organelles and they are vacuoles and lysosomes. So first of all, I will take vacuoles. So what is vacuole? Vacuoles are membrane-bound cell organelles present in the cytoplasm, and they are filled with a watery fluid which is called as cell sap, and this cell sap contains various substances. So vacuoles are present in the cytoplasm of the cell, and they are also membrane bound, and inside the vacuole there is present a fluid which is watery fluid, and it is known as cell sap, and this cell sap, it contains many substances, and the membrane surrounding the vacuole, it is known as tonoplast. It means the membrane of the vacuole is known as tonoplast, and the term vacuole means empty space, and these vacuoles help in the storage and disposal of various substances. They can store food or other nutrients required by the cell to survive, and they store waste products also and prevent the entire cell from contamination. It means that, as you know, that the term vacuole means empty space, and these vacuoles basically they help in the storage and disposal of many substances. They store food or many nutrients which are required by the cell to survive, and they also store the waste products, and rest of the cell, it is prevented from contamination. So this is the role of the vacuoles in the cell. And the vacuoles in the plant cells, they are larger than those in the animal cells, and the plant vacuoles sometimes they occupy more than 80% of the volume of the cell, and the vacuoles may be one or more in number. You must have seen the cell structure of plants, that the most of the portion in the plant cell, it is occupied by the vacuole, and the cytoplasm is present at the periphery of the cell. So in plant cells, vacuoles are larger than the animal cells, and animal cells, they contain small and numerous vacuoles, and plant cells contain few and scattered vacuoles. And the mature plant cell, as I told you, it contains only one big vacuole, and that one big vacuole is formed by the fusion of small vacuoles. Now you should understand that plant cells have vacuoles which are larger in size than in the animal cells, and sometimes the plant cells, they have one big vacuole, and that one big vacuole is formed by the fusion of the small vacuoles, and that big vacuole can occupy more than 80% of the volume of the cell. So this is introduction to the vacuole. So what are the types of vacuoles? First, I talked about the structure of the vacuoles, that it is covered by a membrane which is known as tonoplast, and inside this vacuole there are present many substances, and the role of the vacuole is to store many substances, and to store the waste products also. Then comes the types of the vacuoles. So there are basically four main types of the vacuoles. Number one is sap vacuoles. So it has a number of transport systems for the passage of different substances, and a number of small sap vacuoles occur in animal cells and young plant cells, and in mature plant cells, as I have already told you, that the small vacuoles they fuse to form a large central vacuole and which occupies most of the space of the cell in plants. And the large central vacuole it separates the cytoplasm in the form of a thin peripheral layer. It means in sap vacuoles in plant cells, these are the vacuoles which take or occupy most of the volume of the cell, and the cytoplasm is present on the periphery of the cell, because in the center portion there is a big vacuole, and vacuole is a device to facilitate rapid exchange between the cytoplasm and the surrounding environment. And the fluid present in the sap vacuoles stores many mineral salts and nutrients. So this is about the sap vacuoles. The next type of vacuole is the contractile vacuole. These contractile vacuoles they occur in protist and algal cells found mostly in fresh water. And contractile vacuole has a highly extensible and collapsible membrane. It is also connected to a few feeding canals. For example, in Paramecia, the feeding canals obtain water with or without waste products from the surrounding cytoplasm, and they pour their contents into the contractile vacuole. So, in what is contractile vacuole? Contractile vacuole they occur in protists and in algal cells which are present in fresh water, and the membrane of the contractile vacuole is very collapsible and extensible. So it obtains water with waste products from the cytoplasm, and this water and waste products they enter the contractile vacuole. Then the vacuole swells up, and this process is known as diastole. So when the vacuole swells up, this is known as the diastole, and vacuole swells up because of the water and the waste products. And this swollen contractile vacuole it comes in contact with the plasma membrane of the cell, and then it collapses, and collapsing is known as systole. So what happens when the vacuole swells up? Then it comes in contact with the plasma membrane of the cell, and then this swollen vacuole or the swollen contractile vacuole it collapses, and this process is known as the systole. And then this contractile vacuole it throws the vacuolar contents outside, and so contractile vacuole takes part in osmoregulation and excretion. So what is the role of the contractile vacuole? The role of the contractile vacuole is osmoregulation, means regulation of water content, and moreover excretion of the waste product. So this is the second type of the vacuole. Then the third type of vacuole is the food vacuole. So food vacuoles they occur in the cells of protozoans and several lower animals and phagocytes of higher animals. So these food vacuoles they occur in lower animals, phagocytes and protozoans, and the food vacuole is formed by the fusion of the phagosome and the lysosome. And the food vacuole it actually contains digestive enzymes with the help of which nutrients are digested. So these food vacuoles they contain the enzymes which are helpful in digestion, and the digested material pass out in the surrounding cytoplasm. So when the food is digested, then the digested material passes into the cytoplasm. So this is the role of the food vacuole. So this is the role of these three food vacuoles. Then fourth or the last type of vacuole is the air vacuole, and it is also known as pseudo vacuole or the gas vacuole, and these are present in only prokaryotes, that is the cells with primitive nucleus. An air vacuole or the gas vacuole is not a single entity, neither it is surrounded by a common membrane. So it consists of a smaller submicroscopic scopic vesicles, and these vesicles they fuse to form the air vacuole, and each vesicle is surrounded by the protein membrane and it encloses the metabolic gases. So this is the air vacuole. An air vacuole, it stores gases and it also provides buoyancy, and it provides mechanical strength and protection from the harmful radiation. So what is the role of the air vacuole or the gas vacuole or the pseudo vacuole? Pseudo means false. So these are the names of the same type of vacuole, that is the air vacuole. Either you say air vacuole or pseudo vacuole or the gas vacuole, one and the same thing. And what is the role of the air vacuole? The role of the air vacuole is to store gases, provide buoyancy, giving mechanical strength, and it protects from the harmful radiations. So these are the four types of the vacuoles present in the cell. Then comes the diagram. So this is the diagram of the contractile vacuole in the amoeba. So this is the contractile vacuole in the amoeba, and this is the vacuole in mature plant cell. So this is a big vacuole which is formed by the fusion of the small vacuoles, and big vacuole is present in the matured cells of the plant, and then cytoplasm it moves to periphery of the cell. So these are the diagrams of the contractile vacuole and the vacuole in the matured plant cell. So what are the functions of vacuoles? Now it is clear that what is the function of the vacuole in the cells. So the plant vacuoles, they can store many types of molecules. It can act as a storage organelle for both nutrients as well as waste products. So the vacuole has an important homeostatic function also in plant cells, which are subjected to wide variations in the environment. So it regulates the temperature also in those cells of the plants where the plants are subjected to wide variations in their environmental temperature. And many plant cells, they maintain turgor pressure at remarkable constant level in the face of large changes in the tonicity of the fluids in their immediate environment by changing the somatic pressure of the cytoplasm and the vacuole. So we can say that vacuole also play a role in maintaining the turgor pressure in those cells where the tonicity of the fluids in the surrounding environment changes very quickly, and so they maintain the turgor pressure of the cell. And by increasing in size, vacuoles allow the germinating plant or its organs, for example leaves, to grow very quickly and using up mostly just water. So they also help in growth. So in seeds, stored proteins needed for germination, they are kept in protein bodies which are modified vacuoles. So in seeds there are present stored proteins, and these stored proteins, they are kept in protein bodies. And what are basically these protein bodies? These are the modified vacuoles. So these are the functions of the vacuoles, and this is all about the structure and the function of the vacuoles. Now I will take up next organelle, and that is the lysosomes. Now what are the lysosomes? Lysosomes, it is also an important cell organelle within the eukaryotic animal cells. They are present basically in the animal cells, and they are also known as suicidal bags of the cell. That is the suicide bags of the cell. And this term lysosome, it was given by Christian D. Dubé a Belgian biologist in 1955, and that biologist who discovered it, he got a Nobel Prize in Medicine or Physiology in 1974. So this scientist, he coined the term lysosome, and lysosomes are basically the suicide bags of the cell because of its function. So lysosomes, they are sphere shaped sacs, and they are filled with hydrolytic enzymes that have the capability to break down many types of the biomolecules. So these lysosomes, they have hydrolytic enzyme, that is a breaking enzymes which break down many types of biomolecules. And lysosomes, they are also membranous organelles, and their specific function is to break down cellular waste and debris by engulfing it with hydrolytic enzymes, and that is why they are known as the suicide bags of the cell, because they break down the cellular waste and they engulf it with the help of the hydrolytic enzymes which are present inside the lysosome. So this is a diagram which shows the lysosome. This is the cell and this is the nucleus, and here in the cytoplasm lysosome is present. And if we see lysosome in the enlarged form, so this is the single wall of the single membrane of the lysosome, and inside this lysosome there are present the hydrolytic enzymes or other enzymes. So this is the structure of the lysosome. Then there are different types of lysosomes. Number one is the primary lysosome. What are primary lysosomes? They are formed from the Golgi apparatus appearing as small vesicles. And although primary lysosomes are popular on Golgi apparatus, they also occur as granulocytes and monocytes. And these lysosomes, they are surrounded by a single phospholipid layer, and they contain acid hydrolases. And the pH value of the acid in this vesicle is important in that its changes activate or deactivate the enzymes, and ultimately most of the primary granules will fuse with phagosomes, which results in the formation of the secondary lysosomes. So what are primary lysosomes? These are the small vesicles which are pinched off from the Golgi apparatus, as I have already discussed with you while teaching you about the Golgi apparatus, that vesicles are pinched off from the Golgi apparatus. So those vesicles, they are basically primary lysosomes, and they also occur as granulocytes and monocytes in the blood. And these lysosomes, they are also covered by a single phospholipid layer, and inside it they contain acid hydrolases. So by the fusion of these primary lysosomes, secondary lysosomes are formed. And what are secondary lysosomes? These are formed when primary lysosomes fuse with phagosomes, pinosomes, and they are also referred to as endosomes. They are also called as endosomes. And the fusion also causes the previously inactive enzymes to be activated and capable of digesting such biomolecules as nucleic acids and lipids, among others. And compared to primary lysosomes, secondary lysosomes, they are larger in size and they are capable of releasing their contents or the enzymes outside the cells, where they can degrade foreign material. So basically secondary lysosomes are formed by the fusion of the primary lysosomes or the fusion of primary lysosomes with phagosomes or pinosomes, and then they are called as endosomes. And fusion actually makes the enzyme active, and then these activated enzymes inside the lysosome, they are capable of digesting biomolecules like nucleic acids and lipids. And so we can say that primary lysosomes are smaller in size and secondary lysosomes are larger in size, and they are capable of releasing enzymes outside the cells where they degrade foreign material. So these are the secondary lysosomes. Then next comes the residual body. What is residual body? It is that after the products of digestion have been absorbed by the cytoplasm, then the undigested remains are left in the lysosome, and it is called residual body, and this moves to surface and throws the content by exocytosis. So what is a residual body? That when the secondary rhizosome, it breaks the or digests the bigger molecules, then the products of digestion, they are absorbed into the cytoplasm, and then the remaining portion, that is the undigested portion which is remained in the lysosome, then that lysosome with the remains, undigested remains, it is known as the residual body. And then this residual body, it comes near the surface of the cell, and then it throws its content outside the cell by the process of exocytosis. And the fourth type of vacuole, that are the lysosome, and that is the autophagic vacuole or the lysosome. When a lysosome contains a part of its own cell and digests it, it is called as autophagic vacuole, and it is a part of the normal activity by which old organelles are digested and new ones are formed. So what is autophagic? Auto means itself. When the lysosome contains some part of its own cell and it digests its own cell or part of its own cell by the autophagic activity, then it is known as autophagic vacuole, and this is the normal activity of the cell, because by this process or by this activity the old organelles are digested and new organelles are formed. So this is a type of repair. So these are the four types of the lysosomes. Now what are the functions of lysosomes? So now you know that the function of lysosome is digestion. That is the digestion of large extracellular particles. Then digestion of intracellular particles means digestion of material which is extracellular, and some particles which are from inner side of the cell. So it plays a role of the digestion. And digestion of harmful substances are also taken place by lysosomes, like WBCs. They digest bacteria, germs and viruses to give defense to body. So this is the lysosomic activity. When WBCs, they digest some bacteria, germs or viruses which enter our body, and they make a defense system for the body. So this is the function of the lysosome. So this is all for today. Today we have discussed two organelles, and they are the vacuoles and the lysosomes. So you have to prepare notes. I will send you study material along with this presentation. Then you have to combine the material of both the things. You have to go through this presentation, and you have to note down the doubts if any, and then we can discuss those doubts in the virtual class. Thank you.